Good morning. This is your lecture on cell physiology. So the first few slides we're going to do together in this format rather than cover them in class. So just a little bit of background. As most people are aware, the cell is considered the building block for life. And what a lot of people don't realize, though, is that there are so many different types of cells that each have unique functions. And those functions dictate both the anatomy of the cell and the physiology of the cell. So, for example, what you're looking at here is a white blood cell in the picture. Let's do that. There we go, a white blood cell. And what you'll notice is these are bacteria. And then there are these extensions coming off the cell that allow it to, for lack of a better word, grab the bacteria and bring it up to the surface of the cell to be consumed by the cell. That process is referred to as phagocytosis. The cell consuming a solid substance such as a bacteria. Not every cell has the capacity of doing this. So white blood cells do have the capacity of doing this with these particular cell membrane extensions, but other cells don't. Another sort of example of unique anatomy with a cell are the red blood cells, which in the case of humans have no nucleus. Most cells we're going to talk about do have nuclei, and we'll talk about the function of those, but red blood cells, human red blood cells, do not have a nucleus. So a little bit about very basic cell anatomy. So when you're looking at the cell, the exterior of the cell is referred to as the cell or plasma membrane. And then you'll have a nucleus, and that membrane is referred to as the nuclear membrane. The space in between the plasma membrane and the nuclear membrane is filled with a thick gelatinous fluid referred to as cytoplasm. And within that cytoplasm is where you find the organelles, which we'll talk more about later. The most important thing I want to differentiate right now is what the fluid is referred to in and out of the cell. So the fluid outside of the cell is referred to as the extracellular fluid, abbreviated as ECF, whereas the fluid inside the cell is referred to as the intracellular fluid, abbreviated as ICF. Now both of those, intracellular and extracellular, are made primarily of water. There's other components, of course, but water is by far the largest component of both extra and intracellular fluid. So intracellular fluid is another name for cytoplasm. Another way of saying cytoplasm. The fact that cells are both surrounded with water and filled with water is really important for the structure of the plasma membrane. So before we get into the details of that, there are 4 components of the plasma membrane that you need to be aware of and are responsible for knowing. So there's what is referred to as a phospholipid, we'll spend the most time here in the next few minutes talking about the phospholipid. You also have cholesterol. You have what are referred to as glycoproteins. I just spelled that badly. Glycoproteins and proteins. So if you're asked the 4 components of a plasma membrane or cell membrane, this is the answer. Phospholipids, cholesterol, glycoproteins, and proteins. So a little bit more about the phospholipids. So phospholipids are what it sounds like. It is a lipid that has a phosphate attached. And they're often drawn like this. where this is referred to as the head, and this is referred to as the tail, and the head is the phosphate, and the tail is the lipid part. So if you look at this diagram over here, here's a phosphate, and there are the lipids, phosphate, lipids. And so this is important for the arrangement of the phospholipids in a cell membrane. So if you remember from the little bit of chemistry you reviewed, or if you've taken chemistry previously, lipids themselves are referred to as hydrophobic, meaning water-hating. The phosphate part, on the other hand, of a phospholipid is hydrophilic, meaning water-loving. So if you think about a cell and the fact that you have intracellular fluid and extracellular fluid, that means these phospholipids have to arrange themselves so that the phosphate portion, the lipid, the hydrophilic or water-loving portion, touches the water, and the hydrophobic portion avoids the water. So let's draw a bigger picture of that. So again, here's the cell membrane, and remember there's extra and intracellular fluid. And so if you have a phospholipid, so you have the phospho and the lipid arranged like this, that means the head, which is hydrophilic, is touching the extracellular fluid. But in this picture, we have the lipid portion touching the intracellular fluid, which will not work. And so you have another layer of phospholipid here. They form what's called a bilayer, so that your phosphates are going around the inside and the outside of the cell, coming in contact with the water. And the lipid portion of each of these is able to avoid contact with the water because it is sandwiched in between the phosphates. So it forms a bilayer with the heads facing out towards the water and the tails being kept on the inside here. This arrangement is important for transport of items in and out of the cell, which we'll talk about more in class. And so if you look at this picture here, this diagram, you can see that, appreciate the arrangement. And then down here at this this electron micrograph, you can see the plasma membrane and see how it's got two layers there. So again, the phospholipids are arranged so that the lipid portion does not come in contact with the extra, nor does it come in contact with the intracellular fluid. So the function of the plasma membrane is twofold. One, it helps to separate the inside of the cell from the outside of the cell. So it keeps the cell as an intact unit in and of itself. But it also is designed to allow substances to move in and out of the cell. And the substances that move in and out of the cell do so either through the phospholipids or through the proteins. And we'll talk more about how exactly this works as we move on. So the proteins themselves on the surface of a cell have a lot of different functions. And we're going to spend a lot of time this semester talking about specific different types of proteins. So there are some that serve the function of working as like an enzyme, which we talked a fair amount about enzymes in the last chapter. If you're not remembering, you might want to go back and review that part. There are other proteins that sole purpose is for transport. So the ability to allow substances to move in and out of the cell. And examples of those are pores, also called ion channels. And these specifically allow ions to move back and forth. So if sodium, for example, or potassium were to enter or exit a cell, they're going to move through a pore. And then the other type of transport protein is what is referred to as a carrier. And it moves non-ionic substances. So glucose, for example, sometimes moves through a carrier. Water might move through a carrier. So we'll talk more about these specific types of proteins as we go along. And the other main function of proteins on the cell membrane or plasma membrane are what are referred to as receptors. So I'm going to click this link and we'll watch a short video about an example of what receptors do.
Glucose enters the bloodstream when the body breaks down food to use as fuel. The level of glucose in our blood is controlled mainly by the pancreas, which manufactures and releases the hormone insulin. Insulin works like a key, allowing the body's cells to take glucose up out of the bloodstream and store or use it as energy. In diabetes, two complications can arise. If the pancreas does not release enough insulin, the body's tissues can't take up glucose fully, and glucose levels in the bloodstream rise. The problem is compounded when tissues are insulin resistant and don't respond properly to insulin. Again, the cells can't take up glucose appropriately, and glucose levels in the bloodstream rise.
So let me talk a little bit more about that video conceptually. So the idea in this video is that we'll use the example of a muscle cell. So here's a muscle cell, and it needs glucose in order to perform its function. So you have glucose outside the cell. And glucose I'm going to do like the video did and represent with a little square. And then you also have the hormone insulin that the body makes outside the cell, which they represent looking like a little key. So I'm going to do the same. In order for glucose to enter the cell, insulin has to bind with a protein on the cell surface that is a receptor. So once insulin binds to that receptor, that then opens a passage for glucose to enter the cell. Also through a protein. In this case, it's a carrier protein. So I'm going to repeat that. So insulin's a hormone made by the body. Insulin binds to a protein on the surface of the cell, which is referred to as a receptor. Once insulin binds to that receptor, it then opens another protein, a carrier protein, that allows glucose to enter. So if you don't have insulin, this carrier protein never opens. Also, without the receptor, the carrier protein would not open. So a receptor is a protein on the surface of the cell that binds a molecule, and then that in turn causes some action. The next type of protein I want to briefly talk about, or component of the cell membrane, is what is referred to as a glycoprotein. So a glycoprotein is a protein with glyco attached, or sugar. So they're represented like this. So here's the protein and here's the sugar attached to it. So if you're looking at the surface of a cell, a glycoprotein, you'd have the protein which crosses that lipid bilayer, but the sugar component would stick out into the extracellular fluid. And these are used for cell identification. So, for example, if this were a liver cell, this sugar molecule would help to identify this as a liver cell. If this was instead a red blood cell, there would be a glycoprotein that identified this cell as a red blood cell, and then assuming this person has blood type A positive, there would be a glycoprotein that represents the A, and another glycoprotein that represents the rhesus factor, or the positive. So glycoproteins are used both to identify cell type and then even more specifically blood type. This is also part of what makes you able to get transplants from some individuals. Your glycoproteins have to match up sufficiently to prevent a transplant reaction. If your glycoproteins on your cells are very different, then that's what causes transplant reactions. So here's what we learned in this video. From this video, you should know the definition of the following terms, be able to tell ICF, intracellular from extracellular fluid, know the four parts of a plasma membrane, know the polarity of phospholipid heads versus tails and why they orient the way they do, have some idea about different protein types, and know the function of glycoproteins. In class, we'll have a little quiz over this material and then pick up with the next topic, which is membrane transport.